Oh, good morning, everyone. Good evening. Sorry, good evening, everyone. Oh, sorry. This is the last session. Thank you very much for joining us today. My name is Hideki Taniguchi. I'm happy to have this opportunity to speak to you today. I'm a liver transplant surgeon before, so final goal of my research is generation of human liver. Here, I like to give a talk on development of innovative regenerative therapies for liver disease using human iPS cells. First, I'm going to talk about our research concept, how to make a liver. The second, I want to explain about our status for clinical trials. First of all, I talk about why we must make a human liver. Here is the data from United Network for Organ Sharing. As you know, the absolute organ shortage for clinical transplantation leads to the patient to be dead. In case of liver transplant in the United States, there are 17,000 patients in the waiting list, and death and exit would be 4,000 in a year. To save them, biologists have made effort to develop alternative treatment for organ failure. In this sense, recent great discovery of induced pluripotent stem cells, iPS cells, hold a great promise towards cell-based therapy for such a patient. So many studies are currently aimed to produce mature functional human cells from iPS cells. However, the function of generated cells is still poor. Therapeutic efficacy of cell-based therapy is still controversial, as shown in several clinical trials. Therefore, we aim to generate human organ itself to provide really alternatives for organ transplantation therapy. Today, I would like to give a talk on organoid, represent an intermediate step towards the creation of human organs. This figure listed the developmented human organoid over the past years. Most of the developed human organoids have been used for exploring medical and biological applications. Here is the liver organoid generated in our laboratory. We are now focusing on using the liver organoid for the therapy of liver disease. In the early stage of liver development, we noticed that mesenchymal cells and vascular endothelial cells were present around undifferentiated hepatocytes. So we cultured human iPS cell-derived endodermal cells with human umbilical vein endothelial cells, HUVECs, and human mesenchymal stem cells, MSCs. Notably, although cells were created in two-dimensional conditions, human iPS-derived hepatic endodermal cells self-organized into macroscopically visible three-dimensional cell clusters by intrinsic organizing capacity after seeding. GFP-marked endothelial cells started tuberogenesis and branching to form vascular-like network inside the iPS-derived liver organoid. We examined maturation of iPS-derived liver organoid after transplantation in vivo. Hemodynamic stimulation is essential for liver organoid maturation into the functional liver. This slide shows direct visualization of connection between human and mouse blood vessels in iPS-derived liver organoid transplant. Hideki is labeled with Xavira orange, and mouse-specific CD31 is stained in vivo. Blood flow is visualized with fluorescent dextran. We showed human blood vessels within liver organoid became patent by connecting host vessels at the edge of transplant. As reported, at connected point, human vessels were wrapped by surrounded mouse vessels that called wrapping anosmosis. Transplantation of 12 human iPS liver organoids improved survival of albumin promoter HSV-TK NOG mice. NOG means the IL-2 receptor gamma chain knockout immunodeficient mice after ganciclovir-induced liver failure compared with the sham-operated, the human adult hepatocyte organoid transplanted mice. Thus, successfully generated vascularized and functional human liver by transplantation of iPS liver organoids. These results clearly suggest that therapeutic potential of human iPS-derived organoid transplantation therapy for the treatment of liver failure. We believe that this proof of concept could be applicable to generate functional human liver in clinical settings. To realize this proof of concept for the human patient, we try to establish some challenging technologies, mass-scale production, GMP-grade standardization, and optimal transplant strategies. Organ technology provides a revolutionary paradigm towards therapy, but has yet to be applied in humans, mainly because of reproducibility and scalability challenges. Here, we overcome these limitations by evolving a scalable organoid production platform entirely from human iPS cells. Because transplantation of at least 10 to 8 or 10 to 9 hepatocyte is required to correct specific metabolic liver function in human. Now, we are preparing a new GMP-grade cell processing facility for clinical trials. We call them CPWS, cell processing workstation, considered of stock unit, interchange unit, isolators for cell culture, and semi-automatic incubators. This is the semi-automatic incubators. These special incubators contain over 100 culture plates. Inside the incubator, the robotic arm carries the culture plate to the connecting box. The operator orders to bring the white box from the stock unit. Each white box contains bottles of medium, pipette, culture plate, and prepared human cells. The white box runs through the interchange unit and stops in front of the isolators for cell culture. The operator opens the door to take the white box inside the incubator under the strictly clean conditions. The robotic arm guided the plate to the cell observation module, and the operator can observe the cell in the CPWS monitors. We achieve human scalability by developing an all-new well array culture platform for mass-producing homogeneous and miniaturized small liver organoid on a clinically relevant large scale. Fluorescence image analysis confirmed the large-scale production of small liver organoids from human iPS cells. When we use this micro-pattern culture plate, we can produce large amount of size-controlled homogeneous human liver organoid from iPS-derived cells. Here is the SOP workflow of human iPS liver organoid manufacturing. To ensure the stability of liver organoid manufacturing, we established a master clinical grade human iPS cell bank in the cell processing facility. Next, we differentiate the human iPS cells into hepatic endosome, endothelial cells, and mesenchymal cells with our standardized culture protocols. These processes take about 24 days. For liver organoid generation, these three types of cells are mixed at an appropriate rate and seeded in a three-dimensional microculture system. With another 8 days of differentiation, we get the matured liver organoids. Throughout the manufacturing process, we set a series of quality control standards to ensure the quality of the final product. Here is the morphology of the liver organoid. After 8 days of differentiation, qPCR showed that liver organoid have a much higher maturation stage than human iPS cell-derived hepatocyte. Improved function of liver organoid were also confirmed with ammonia metabolism and urea production and lysis. We also set quality control standards. As shown in this slide, the liver organoid generated in a standardized protocol had extremely high qualification rate. So we can use these iPS liver organoids for clinical trials now. Recently, we reported our third generation liver organoid, which are large in size and exhibit significantly improved structures which have a similarity to fetal liver tissue. These new organoids can be created by the fusion of conventional our iPS liver organoids and have disc-like structures. Here, we present the morphology of our third generation organoids, which are approximately 20 times larger than conventional iPS liver organoids. Within these organoids, we observed the formation of capillaries and small vessels surrounded by the Vimentin-positive pericyte, as well as bile duct structures resembling fetal liver tissues. Now, let me show you the time-lapse imaging of the fusion process of organoids in detail. As second-generation small iPS liver organoids are fused together within a mold, theoretically allowing for unrestricted scalability in size. iPS-derived hepatic endosome cells, you can see in green color, make small spheroid-like structures. On the other hand, endothelial cells, you can see in red color, start to form vascular network between green color small conventional liver organoids. We consider our new approach could potentially be applicable to MASH, metabolic dysfunction-associated steatohepatitis. To realize them, we have performed additional experiments for clinical application of human iPS liver organoids. Currently, MASH have become a significant social issue worldwide. In the United States, more than 30% of adults are affected by MASLD, metabolic dysfunction-associated steatotic liver disease, which serves as a precursor to MASH. Moreover, the patient number is continuously increasing. MASH is a progressive type of MASLD, accompanied by liver fibrosis. approximately 20% of monthly patients rapidly develop liver fibrosis with 10 years, with an increased risk of hepatocarcinogenesis, and 20% of patients with liver fibrosis progress to cirrhosis within 2 years, potentially resulting in liver-related deaths. Therefore, inhibition fibrosis progression is considered crucial for establishing effective therapeutic strategies against MASH. In developing the third-generation liver organoid for MASH therapy, we first investigate whether fetal liver tissue can be used as a suitability graft for transplantation. Here, we develop a new transplantation protocol which helps transplant the rat fetal liver tissue onto the liver surface of the liver failure model. Histological analysis revealed the presence of CD31 positive blood vessels, SE1 positive liver sinusoidal and cellular cells, and CK19 positive bile duct cells within the engrafted tissues. More importantly, we noticed the transplantation of fetal liver tissue demonstrated a significant improvement in survival of liver failure model. Here is the development process of fetal liver. The size of the fetal liver increases from the embryonic 9.5 stage with a remarkable angiogenesis. On the basis of these findings, we generated hundreds of small iPS liver organoid on micropattern plate and then cultured at high density on cell culture insert, which helps enhance the static cellular interactions and induced angiogenesis. In this organoid, we observed the formation of capillaries and small veins, the blood vessels surrounded by hepatocyte, the presence of space of disc exhibiting morphological characteristics similar to fetal liver. As you can see in movies on the right upper panel, iPS liver organoid generation process involves formation of remarkable network of vascular and cellular cells. Moreover, the novel iPS liver organoid exhibit increased expression of hepatic functional related genes when compared to our conventional liver organoids. The enhanced hepatic function of this liver organoid were further confirmed with production of human albumin and transferrin and metabolism of ammonia. Based on these findings, we concluded that novel iPS liver organoids exhibit superior similarity to liver tissue and possess excellent liver functions. Next, we investigated the tissue construction ability of third-generation iPS liver organoids in vivo. After 2 weeks of surface transplantation, human albumin-positive hepatocyte and CK19-positive bile duct could be detected in the graft. Bile duct structures were observed near the vascular structure within the graft, which resemble the portal area of fetal liver. The engrafted hepatocyte exhibit mature hepatocyte ultrastructures, including glycogen granules, mitochondria, and RAF and plasmic reticulum. Moreover, we intravenously inject fluorescence-labeled human-specific anti-CD31 antibodies into the transplanted mice and observed well-developed human vessels formed in the engraftment. Additionally, iPS-derived bile duct formed within the graft exhibit specific features such as primary cilia and microvilli on the apical membrane and transporting carboxy dechlorophorescent CDF into the bile duct lumen. Notably, the transplanted iPS liver organoid could stably engraft it in vivo with a stable detection of human albumin, human complement factor C3. These findings suggest that the transplanted iPS-derived liver organoid can be matured into functional liver tissue in vivo. We then investigate the therapeutic effect of third-generation iPS liver organoid in treating liver failure. As shown in this slide, transplantation of iPS liver organoid into rat liver failure model demonstrated significant improvement in survival rate. Compared to the sham group, the transplantation group showed reduced ammonia level, increased platelet count, and decreased prothrombin time. Moreover, the decreased level of liver injury marker, including AST, ALT, and bilirubin, were observed in the transplantation group. These findings indicated that iPS-derived liver organoid transplantation significantly ameliorate the condition of liver failure. We noticed that the third generation iPS liver organoid were high detection of genes related to macrophage, migration, and polarization. Fetal liver is the site of hematopoiesis, as you know. Our fused iPS liver organoid release a lot of cytokines and chemokines, which have some effect to the blood cells. Cytokine RA analysis further confirmed that iPS liver organoids express macrophage migration factors such as CSF1 and CXCL12, as well as anti-inflammatory M2 macrophage inducing factors such as IL-4, IL-10, IL-13, and HGF. Moreover, transwell co-culture experiment revealed that iPS liver organoids possess the ability to induce macrophage migration. This migrated macrophage had upregulation of M2 macrophage-related markers. Additionally, these co-cultured macrophages were found to highly express anti-fibrotic factors MMP8, 9, and 13. These results suggest that iPS-LO liver organoid could recruit macrophages and further induce macrophage polarization toward M2 macrophages. We investigate whether transplantation of iPS liver organoid exhibit therapeutic effect against liver fibrosis. Compared to sham group, iPS liver organoid transplantation significantly improved the survival of mouse within 6 weeks post-transplantation. Serious thread staining revealed that iPS liver organoid transplantation resulted in alleviation of fibrosis. Moreover, iPS liver organoid transplanted liver showed an increased detection of FO80 positive macrophage. To further confirm the role of macrophage in fibrosis resolution, we administered chloridronate liposome to inhibit macrophage function in iPS liver organoid transplanted mice. Notably, chloridronate liposome administration diminished the therapeutic effect of iPS liver organoid transplantation on liver fibrosis with a decreased population of CD168 positive phagocytic M2 macrophages. These findings suggest that iPS liver organoid could regulate macrophage recruitment and polarization to improve liver fibrosis. As mentioned at the beginning, organoids are intermediate step towards the creation of fully functional human organs. We believe that complete organs are the best candidate for clinical application. Therefore, we are actively working to further advance organoid technology. Currently, we are establishing liver organoids that possess large blood vessels and bile duct using human iPS cells. Hemodynamic stimulation is essential for liver organoid maturation into the functional liver. We are working on establishing in vitro culture medium perfusion system for liver organoids with iPS-derived large blood vessels. As shown in movies on the right lower panel, when the iPS liver organoid with large blood vessel is anastomosed surgically with blood vessels of recipient animals, blood flow was confirmed not only in the large blood vessels, but also inside the iPS liver organoid without causing leakage of blood. As you can see here, after the removal of stopper, blood was supplied into whole liver organoid through a large vessel. Notably, we found that the transplantation of this liver organoid with large blood vessels could improve the mice survival from liver failure. We will continue to move forward to realize next generation regenerative therapies by creating human organs, which can be surgically transplanted through vascular anastomosis. I appreciate all my colleagues of this work. I also appreciate my collaborators, Dr. Higashibata and Uemura in JAXA, and Dr. Suematsu and Dr. Hideki in Keio University, and Gray and Barber in Max Planck Institute. I'd like to express my gratitude to all of you. It has been a great pleasure being with all of you today. Thank you very much for your attention.